Okay, all right, so here we were talking about haploinsufficiency as soon as it pops up here. And haploinsufficiency is just when you have what would be a recessive allele on one chromosome and a wild type. But there's a difference. So essentially haploinsufficiency is the same thing as incomplete dominance. So in this case, it's a product being made, and so you don't. There are some mutations of Notch that are not going to work in their intended function as a receptor. And so, like I said before, if you completely knock out Notch, then you're dead. But if you have Notch over plus, it's haploinsufficiency. So you have enough of the receptor, enough of the good receptor around that you make a fly and you go through development okay, but you get these little defects, the little notching in the wing there. All right, so now we're going to go through the inversions and translocations that we talked about in the second lab. So this is where when you're studying, you should go back and look at all the pictures you took, which should hopefully help you understand this. So we talked last time, and there was a question about what's the difference between a reverse duplication and an inversion. Here, and I explained it last time a little bit, and here's the inversion to show you. So in a reverse duplication, now it just involved E and F, but let's say it was D, E, and F. If you had a reverse duplication, you would have the normal DEF, and then you would have a flipped DEF like this stuck in somewhere else. An inversion is when part of a chromosome just flips and goes back together. So what causes this? All kinds of either chemical or radiological, gamma rays that are just around, X-rays, things like that, can cause breakages in the chromosome structure. So what happened was a break of the DNA here and here occurred. There are many different pathways that the cell uses to repair these breaks, the most common of which is that the cell will look at the other chromosome, which isn't shown here, the wild type version of the chromosome, the homolog, and figure out how to make these repairs. But in a number of cases, those mechanisms don't work, and the cell needs to go through cell division, and the cell's facing a choice. Either slap things back together as well as you can and see what happens, or you're going to die. You're going to have to go to apoptosis because you can't continue to go through the cell cycle. And so what happens is it just jams everything back together. Now, if there was a single break like right here, and you were lucky, it would just tag this piece right back on to the ABC piece there. And there might be a little bit of a problem with DNA sequence right where it goes back together because it wouldn't be perfect. But the chances that you hit something important, like a gene or a gene promoter, the switch region that we talked about, is pretty low, given that there's a lot of DNA that doesn't really code for anything in our genome. But here, you've had two breaks, one here and one here, and instead of getting put back together in the correct orientation, everything is flipped. And so now the genes are facing the opposite orientation of that they were before. I mean, it's these are what's wrong, what's happening is these are upside down and they are backwards there. So check to make sure in your book. I'm not sure, I think the new books corrected it, but I think in the old books they had like the F, the E, and the D being underneath but upright written like this. They would be facing the other way like I have shown here. This is correct in what it should be. So the problems that happen here are not what happens with with duplications and deletions? Well, the first one isn't, because you're not adding any sequence or getting rid of any sequence. If everything goes okay, you've got the same genes here that you do up there. The only issues that you see with this kind of an event, at least before we talk about crossing over and recombination during meiosis, is that if something got broken here or here, where the chromosomes got broken before the flip happened, like if there was another gene here that was split in half, or if the break happened right here and now one of the genes that's in the area of D is separated, like its promoter or switch is still over here, and now it's either not turned on at all or falls under the promoter of another gene where it would be turned on at times it's not supposed to be, you get these position effects. So position effects are going to be important for inversions and for translocations. All right, so let's go back and talk about what we talked about in lab on the second week of lab. So we're going to talk about paracentric inversions and pericentric inversions. So you're going to have to know the difference between them, and I usually try to give some small amount of leniency if you don't spell a word perfectly correctly, but if you spell paracentric and pericentric incorrectly, I can't give you credit because they're two different things. So paracentric is not involving the centromere. So here's the normal chromosome, here's the inverted chromosome. You just have this flipped upside down and backwards. But the centromere is not involved. So everything from here to the left and everything from here to the right in the inverted chromosome is just as it should be. As I asked on the homework for that lab, a paracentric inversion, if it becomes homozygous, is not a problem. So if you are homozygous for an inversion, now, okay, so we're assuming here that there's no position effect problems from flipping these around. Otherwise, the organism, wouldn't be alive. So we've got a homozygote right here, these two inversions, and now, even though both of these genes have this inverted part compared to the wild type chromosome, both of these chromosomes are exactly the same. And so you can have recombination in a paracentric or pericentric inversion homozygote, and it's not going to cause any problems. It's just like recombination anywhere else. Why is that? It's because the problem comes when you try to pair in an inversion heterozygote, where one of the chromosomes has a wild type sequence and the other one contains an inversion. The chromosomes want to pair up. That happens by looking at homologous sequence. So these sequences right here are all the same. Here. These sequences here are all the same, so they can line up, but these are different. What happens then is that either the paracentric part or the wild type part, one of them, you put them together like this, and one of the two has to have an inversion loop forming like that. And so DNA is very flexible, and this isn't a problem. So we're going to, chemistry alert, the lowest energy state here, right? And that is you want to pair the greatest amount of the chromosomes possible. And so this preferentially forms versus just having this bulge out. We talked about having duplications or deletions. You get a bulge forming when pairing happens. That's because some of the sequence isn't even there. The inverted chromosome has the C, D, and E sequence like here, but it's in the opposite orientation, so it just flips around like this. If you get crossing over and recombination anywhere outside the region that's inverted, So anywhere from here to the left or from here to the right, there's no problem because you're just exchanging this piece of this chromatid with this piece of that one, or the part that has the F and G here with the part that has the F and the G over there. No big deal, right? The problem is when you get a recombination that happens after crossing over within the inversion. And so remember in the last chapter we talked about the recombination and how to follow the chromosome with your finger from the left to the right to see what happens. That works with this as well. So because we're having these flip apart, this one up here is still by itself, right? That's the one that's not inverted. And this one down here is fine. It has the inversion, but this one and this one are having a problem because they're the ones that had the crossing over. So follow this. You're going to end up with one dicentric chromosome that looks like this. Dicentric because it has two centromeres, and then an acentric. So here's the one where you get the dicentric chromosome, so you follow this around. Now right here, this was the original part of that chromatid, but you have crossing over, so now you're going here and then back down there. So there's one centromere, there's the other one. And this one here is, oops, that's the same one. So you follow this around to there, and then where's the acentric one? It's this guy right here. So you follow this around underneath there and around down here. That's this acentric chromosome. So centromeres, remember, are required. Centromere is DNA sequence to which proteins that form the kinetochore bind. And that's what the microtubules attach to move the chromosomes during meiosis or mitosis. So forming the acentric chromosome here, that's going to all get lost. All that DNA is just going to get chopped up into individual nucleotides, and it's going to get reused by the cell. The cell will recognize that the chromosome doesn't have a centromere and destroy it. The dicentric chromosome is going to have a problem as well, because you still have on the left-hand side here, the part that hasn't undergone the recombination, you still have part of one homologous chromosome here and the other homologous chromosome here. So what happens at anaphase of meiosis 1? The homologous chromosomes separate. So this one's going to want to go up and this one's going to want to go down. They move apart from each other. And that's going to be a problem because this sequence here is all hooked together. You can actually see this in metaphase chromosome spread. So here's half the homologs going down, half the homologs going up in this picture and in that one. And right here you see the dicentric bridge. Now the DNA is held together by covalent bonds, but those covalent bonds holding the sequence, the DNA chains together, are not stronger than the forces exerted by the microtubules pulling on the kinetochores. So somewhere in here and here, somewhere in this sequence, the DNA is going to give and it's going to get ripped into two pieces, and it's just sort of random where that happens. So what happens here is the break is going to happen about right there, but it's just going to be somewhere. So you end up with this piece ripping right there, and this goes to the top chromosome like that, and this piece goes down with the bottom chromosome. Now imagine that these are like, this is cut off here, this one is folded down there, and then these two chromosomes are going to line up at meiosis 2 metaphase plate, and the sister chromatids are going to separate. When the sister chromatids of this top one separate, you get one normal sequence chromosome, and this one down here which is a piece of the dicentric bridge. On the bottom, you get one complete chromosome that has the inversion and the other piece here. So these are big amounts of DNA that gets lost here, and you have the dosage effect again. So you end up with this gamete is missing all of these genes right here. So when it gets fertilized by a normal gamete, let's pretend this is the normal gamete and this is in the other one, you're going to have two each of the red, orange, and yellow, but the green, blue, violet, and gray are going to be missing. So you'll have two doses of these, only one of those, and that dosage imbalance is going to make the embryo kaput, inviable. It's going to die as a ball of cells after only a few cell divisions. Same with this one down here. If you have this chromosome fertilized by a normal gamete, it's going to have two of the red, orange, green, or blue and green, but it's going to be missing the yellow, purple, and gray here on the end, and same thing. So half the gametes are going to be viable and half the gametes are going to be inviable. Remember, this is the same thing that happens, not the inviability part, but it's because half of the gametes are recombinant and half are not. So this one and this one are the non-recombinant gametes, and these two are the ones that underwent the recombination. So the recombinant gametes are inviable. These two are fine. The reason why they're inviable is because they're all the recombinant gametes are missing sequence that was lost to the acentric chromosome. Peri means around. Pericentric inversion includes the centromere, so you can see a break happened here to the left of C and there to the right of E. This piece gets picked out, flipped around all the way, and right there. Because the centromere is included in the inversion, you're going to have a difference in the gametes that come out of this. Again, same thing. There's no picture, but if you had a homozygote of a pericentric inverted chromosome, any recombination would be fine because the chromosomes are the same. Here, oh, and also, just like with the pericentric, if you have a recombination from here to the left or from here to the right in the parts that don't involve the recombination or the inversion, then recombination there is going to be fine as well. All right, so here we're going to have a crossing over and a recombination event. And in this case, you're not going to lose any sequence. There's no acentric chromosome. But what's going to happen is you're going to have unequal swapping of genes between the two non-sister chromatids. Let's follow this one around. So this one's going to have red, orange, yellow, green, blue, and instead of purple and brown or gray, you're going to have another orange and red. This one is going to have okay, so it's this one they're using. Well, no, we can use this one. This one's going to have the brown or gray, the purple, blue, and then green, and then yellow, but instead of orange and red, you get another one of these. So essentially, the orange and red and the purple and brown or gray there has been swapped between them. It's easier to see when you look in the next picture here. Now when these two separate, this chromosome's going to get pulled toward the floor, this one's going to get pulled toward the ceiling again. There's no dicentric bridge. This is not covalently bound, this chromatid is not covalently bound to that one. They're just held together by cohesins, which break when the chromosomes want to separate from each other, and it's fine. So when they get pulled apart, you end up with this. One normal and one inverted that have everything, all the genes, and then each of these in the middle is going to have a duplication and a deletion. This one has a duplication of the F and the G region and a deletion of the A and B. This one has a duplication of the A and B and gotten rid of the F and the G region there. The same thing happens regarding viability. With the para and pericentric inversions, you're going to get half of the gametes non-viable in each case. But unlike the paracentric inversion, in which there was sequence lost so that all the non-viable gametes had sequence gone, here the non-viable gametes are each going to be missing some sequence and got some more sequence there. So let's look at this one. If this gamete gets fertilized by a normal gamete, let's just use this one as an example, you're going to have 3 copies instead of 2 of the purple and brown there. And you're only going to have 1 of these. So you get this gene dosage effect again over a big area. You've got too many of some things and not enough of others. And the reciprocal is true for this chromosome. You're going to have 3 of the red and orange and only 1 coming in the normal gamete of the purple and brown. These 2 gametes on the outside were not involved in a recombination, and so they will both be viable. Even though this one has the inversion, just by existing as an inversion heterozygote, you know that this break here and this one here on either side that led to the inversion could not have disrupted any important gene, because otherwise it wouldn't be able to exist at all. Good so far? Yes. I'm not going to make you draw the loop-de-doos on the exam, but you should know what happens in each case, right? I'm not super good at drawing either, and what happens when a long time ago when I did ask that, people take so long doing that on the test that they don't have time for anything else. There will be questions about all this material, and you will have to know what happens. I'm just not going to make you draw like this, right? You should know what happens and know that this is important. So I may not ask you to draw this exact structure, but what's important to know here is that neither of these is an acentric chromosome. So there's no dicentric chromosome and there's no acentric chromosome, so that when these pull apart, you get no breakage like you do here, breakage of the dicentric bridge. Yeah. Yeah. Yep, that's exactly right. So what happens in paracentric is you get the acentric chromosome and that sequence is lost. So each of the recombinant gametes has lost some of its sequence. Here, no sequence is actually lost. You're getting exchange of DNA sequence between the homologous chromosomes, but it's not homologous sequence, right? So this one, so up here, so this chromosome, either or. So one of the chromosomes is going to end up with an A and B at both ends, and one's going to end up with an F and a G at both ends, and you can just look at this and follow it around. like that, right? So if recombination didn't happen, you would have A, B, C, D, E, F, G, right? But because you have recombination within the pericentric inversion, you get A, B, C, D, and then E, and then B and A again. And the other one gets the opposite. So this one is going to have the purple and the brown or gray at both ends. There they are at one end, and here they are at the other end, because this recombination keeps it from going all the way through. You're getting exchange of sequence. So one of the chromatids is going to give the red and orange to the other one, and that one gets back the purple and the gray. And it's just a function of the fact that there's that pericentric inversion that is present there, right? All right. So what would happen if you got a double crossover event within a pericentric inversion? What happens if you get a double crossover within two genes? The first crossover and recombination would switch from one chromatid to the other, and the second one would switch it right back, wouldn't it? So if you get a double crossover event taking place and they both have recombination, two recombinations within either the peri or the pericentric inversion, the first will lead to trouble, but the second will put things back the way they're supposed to be. And because they both happen within the inversion, then everything comes out okay. So if you have recombination anywhere in an inversion homozygote, you're fine, because essentially there's not a difference. If you have recombination anywhere outside of the inversion in either a para or pericentric inversion, so over here to the left or over here to the right, outside of the inverted region, no big deal. The problem arises when you have a recombination in the area where the inversion loop exists, because you get the DNA flipped around backwards. Then you have a recombination and it exchanges the sequence and locks it together. Good? All right. So, as with everything else that we discover in genetics, the scientists actually use these things, and the fact that recombination between an inversion and a non-inversion chromosome leads to inviable gametes for our own purposes. So we have on a lot of the C. elegans chromosomes inversions that have either naturally been found, or most likely we take the C. elegans worms, put a bunch of them on a plate, and stick them in very close proximity to gamma radiation. And instead of, you don't get excited, there's no Incredible Hulk worms. They don't turn green when they get angry. Instead, you get a bunch of inversions, and most of them are dead because bad things happen. But occasionally, you get one where the inversion didn't mess anything up. This is chromosome 1. So over on the right-hand side of chromosome 1, there is an inversion where this part in dark blue has been inverted there. So you can see the UNC75 and UNC54 genes have been inverted are in the opposite orientation there. We call this one on the bottom here a balancer chromosome, because any gene on a normal chromosome within the part that's inverted will be balanced, we say, by that chromosome. You're able to maintain it. I'll show you what I mean. So one of the genes that I'm looking at is called FER14 in my lab. It's another sterile mutant. FER is just fertilization defective. Think of it as the SPE, spermatogenesis defective, same thing, right? So FER14 is in cis to UNC54. So this is just sort of like a paralyzed UNC, just like you've seen in the lab. So if you have this over a wild type chromosome, that will be wild type because you're heterozygous for both, and then 1/4 of the progeny will be homozygous for this chromosome, and you look for paralyzed worms and they'll also be sterile. Ta-da, right? The problem is that even though these two genes are relatively close together, recombination can separate them so that if you do this over a long period of time, some of the paralyzed worms are no longer going to be sterile and you've lost your gene. So sterile genes or lethal genes, you want to be able to keep them and you have to maintain them as a heterozygote. If you're homozygous for FER14, you can't make any babies. If you're homozygous for a lethal allele, you're dead. You don't even develop. So we're going to balance the chromosome that has UNC54 and FER14 in cis to each other over this balancer that has the area between these genes flipped. So this chromosome will have UNC54 and FER14 wild type genes on there because its only mutation is over here. So not only does this have an inversion, but remember there's UNC75 gene and UNC54 gene. We talked about different alleles having different phenotypes. There are some alleles of UNC75 that make the worms paralyzed, but if UNC75 is completely not there, it's lethal. So this inversion also has a lethal allele on it, right? So this heterozygote looks, walks, and talks like a wild type worm. Because this chromosome has UNC54 and FER14 mutations, the balancer has wild type genes, everybody's okay. The balancer has a lethal mutation here, but it's recessive. Here, there's an UNC75 wild type allele that I just didn't draw in. If you get a homozygote of this one, you get paralyzed and sterile. Yep. That's exactly what's happening. That's exactly what's happening. The two mutations that I'm trying to maintain are complemented by the inversion allele. Meanwhile, the homozygous lethal here is complemented by this guy up here. Right. Yeah, they're exactly, this is one homologous chromosome, this is the other one. Everything in blue is exactly the same between them, only the part that's in, well, the light blue, only the part that's in dark blue or purple is different there between them. Because these are the same chromosome, you can think of this as one allele and this as another allele together, right? So busy slide, but that's why I wanted to introduce everything here. So, oh, whoops, never mind, I have this one first, sorry. If you cross this one to this one, and really we're not doing a cross, we're maintaining it in a hermaphrodite, so it's the sperm and the egg are the same thing. Here are the possible gametes, just like what we showed on the previous page. And if you look at, this is the busy slide, if you look at a Punnett square, this is it, right? So 2 out of 4, you're going to get one non-inverted chromosome with the mutations I want to maintain, one chromosome with the lethal mutation and wild type for my genes there. These two will be, I put wild type, they're not, they have a lot of mutants, but these two will be not paralyzed and also not dead and not sterile. And then if you get homozygous for this one up here, some of the worms will be paralyzed, and they'll also be sterile. So if I want to pick sterile worms off this plate, all I got to do is look for, if I want to pick sterile worms off the plate, all I have to do is look for the paralyzed ones. And then down here, anybody who's homozygous for the balancer chromosome is going to be a sad little egg on the plate that never hatches. So this heterozygote that looks like this is going to give 2/3 of the same thing to keep the line of worms going, and 1/3 of what I want. So it's the gift that keeps on giving. And because the balancer chromosome is lethal, I don't have to worry about that. Some balancers are not lethal, and they'll have like a dumpy or something in them, so that I have to be sure to keep picking wild-type worms. But here I don't have to worry because this guy's dead. And any recombination that wants to happen, so here's UNC54 and FIR14, there's 2 wild types there. So if this was not an inversion, you could get recombination between here and unhook the paralyzed UNC54 from FIR14. But anybody who has a recombination event between those two, they're dead too. And so it's an automatic thing that just keeps going. Did you guys have another? Everything okay? All right. So we've taken this inversion and used it in the lab to maintain either sterile or lethal alleles. I'm not going to show it. Yes? What happens in the wild? Let's talk about inversions first. If you get an inversion, then that would not last in the population, because being an inversion heterozygote means that anytime you get recombination in there, those gametes are you're producing a certain fraction of inviable gametes. Even if it's a really small fraction of your gametes that are inviable, if everybody else in the population's gametes are 100% viable and yours are 99.5%, that's going away. That's a strong enough selection in a population that after a while it's going to go away. Other alleles, like a sterile allele, if you're a C. elegans worm and you get a mutation that happens where you're now heterozygous for a sterile allele, that means 1/4 of your progeny will be sterile and not make any babies themselves. Well, you know, evolution puts the big thumb on that as well, and if everybody else is making 100% progeny that will have progeny of your own, and if 25% of all the babies you make don't make any babies themselves, that's gone. I mean, it'll take many generations to happen, but it all gets weeded out. This is all artificial, and we're generating these mutations in the laboratory because we're trying to study, in this case, the process of making sperm and sperm fertilizing eggs, right? And so we have to be very careful when we generate these mutations to immediately try to get them preserved like this, or all the work you put into making the mutations is gone. So we have these systems set up. Yeah, that's why it's really important to plan out your experiments. So we use this to maintain alleles, and when we talk, I'm not going to show it, but when I talk about the translocations, those are also a problem, right? Where you'll see that half of the progeny are going to be inviable if you get, well, we'll see. Half of the progeny are going to be inviable because you get one segregation versus another at meiosis 1. Those can be used to maintain sterile alleles too. All right. And yeah, like I said, if you get any recombination in there, they're dead. So you're done. So a translocation is a similar issue where you get a break. So in an inversion, you get a break at 2 spots in one gene, and then everything gets flipped and put back together. With a translocation, in this case, you had an original chromosome that looked like this and suffered a break here, and another chromosome that looked like this and suffered a break there, right? Now these are duplicated or replicated chromosomes. So when the break and repair happens, they're not going to be looking like this. They're just going to be like a single line, like one of these chromatids. And that repair mechanism that I talked about that says, uh-oh, something really bad's happening, I better jam these back together or the cell's going to have to die. What happened here is the piece that broke off the blue chromosome gets stuck onto the red one, and the piece that broke off the red chromosome gets stuck onto the blue one. Again, you're subject to position effects here. If the spot between D and E that broke was in the middle of a gene, wouldn't be any good because it would be trouble. You wouldn't develop. Or if the E gene here had a promoter there, and now the promoter is either gone or it's got a different promoter, so it's regulated differently, that position effect issue, same thing. So we're assuming that the area where the breaks and repairs happened don't contain anything that's necessary for survival, or you wouldn't get this. So now you've got a translocation heterozygote. The red ones on the left, so the left part, just look at the left part here. These were two homologs, and these two were 2 homologs too. You can see the 1s and the 2s there. You can think of the red one as chromosome 1 and the blue one as chromosome 2. So if this didn't exist, at meiosis 1, you would have the red ones lining up as a pair, the blue ones lining up as a pair, and then the homologs would separate. This centromere separates from this one, this centromere separates from that one. But now you've got these pieces on the end. However, the part of the centromere that does not contain the inversion still tells the chromosome who it used to be. So these guys know that they're number 1s, and these guys know that they're number 2s. When you get pairing during meiosis 1 prophase and metaphase, instead of that inversion loop that forms with the inversion, here you get a cross-like structure. That allows the biggest part, like here, and these two here, so it allows all of the chromosome sequence to pair that is able to. That's what it wants to do, and so this just happens. Right? The centromeres remember who they are, so at anaphase of meiosis 1, one of the centromere number 1s is going to go one way, the other one's going to go the other way. One of the centromere 2s is going to go one way, one's going to go the other way. Just like if the translocation didn't happen here, which way they go is random. So that's why there are 2 different segregation pathways that can happen. I know there's 3 shown, but let's hold off on that one for just a second. So if you look at this one called alternate, the corners go. So this chromosome here, N1, and this one here, N2, go to one daughter cell, and T1 and T2 go to the other one. So here's one daughter cell after meiosis 1. Here is the second daughter cell after meiosis 1. In adjacent 1 segregation, you split it here, and the two on top go up and the two down below go down. So you have an N1 and T2, T1 and N2 there. In both of these two cases, the left and the middle, each daughter cell got one chromosome 1 centromere and one chromosome 2 centromere. The N and the T are just to notate normal and translocated at this point, right? So alternate because you're getting the corners, they're on opposite sides of the cross structure, and adjacent because they're going this way and that way. We're going to come back to that one. In alternate segregation, all the gametes are viable. Think about it. What do you need? You need one copy of each gene. So here, in alternate, this cell is easy. It's got, N1's got one of each of the red genes, and N2's got one copy of each of the blue genes. So when you fertilize those gametes, you'll have two of everything, and we're all set. Even though these two translated chromosomes look funny, they serve the same purpose. Between T1 and T2, you have, look here, this plus this is all of the red genes. This plus this is all of the blue genes. Again, this is based on the fact that the spot where the translocation happened didn't screw anything up. Yeah. There is no complementation here, because complementation is where you have two homologous chromosomes and a mistake in one is made up for in a wild type gene in another. In this gamete, you only have one of every gene. Here's one of all the red genes. Here's one of all the blue ones. Look, you only have one copy of A through G and one copy of M through S. These two are not homologous chromosomes. They're completely different. This one here? Yep. Okay, look. Still, that's down here. You still only have one of A, B, C, D, E, F, G, right? You still have only one gene each of M, N, O, P, Q, R, S. So you're thinking, I can't let you say complementation because it's a specific genetic term and this isn't it. But yes, between the two chromosomes, the piece that's missing from this single chromosome is present on that one, and the piece that's missing from this one is present there. This one is missing E, F, G, but it's present there, so we're good. This one is missing Q, R, S, but it's present there, so we're good. Between these two translocated chromosomes, they have one of every gene. Exactly the same as you see with this. So all these gametes are viable. So let's compare that to what happens with adjacent one segregation. In each of these cases, let's do these two in the middle because they're the differences. Look, this one has one each of all the red genes on the N1 chromosome, but this chromosome down here has the E, F, and G part. So this gamete has two of E, F, and G, but it's missing the Q, R, S. So it's got two of some things and zero of others. So when this gets fertilized, it's going to have 3 copies of E, F, and G and only 1 copy of Q, R, and S because there's none in here. So that difference in dosage is going to be, that dosage effect is going to mean that is an inviable gamete. Same thing, but for the opposite reason down here. This one got an N2 chromosome that has MNOPQRS, everything's fine, but this chromosome, the translocated one, has A, B, C, D, but is missing E, F, and G. So this one, when the gamete gets fertilized, because there's 2 of Q, R, and S already, now there's going to be 3. But because there's no E, F, G, when you add a normal chromosome with just one of those, it's only going to be 1 instead of 2. Does that? That makes sense? Everybody else? Questions? I'm not seeing any questions, but I know you don't get it, so let's have some questions about why not. All right, so this has adjacent to segregation circled and it says rare. Why is it rare? Let's go back to this one again. What happens in meiosis? 1, anaphase. The homologous chromosomes separate. What happens if 2 homologous chromosomes do not separate but instead go to the same gamete? It's non-disjunction, right? So in adjacent to segregation, you're going to draw a vertical line here, and the 2 chromosomes on the left are going to go to one daughter cell, and the 2 on the right are going to go to a daughter cell. Do you see the problem here? These are 2 of the chromosome 1s, and these are the 2 of the chromosome 2s. So in both the left and middle case, each daughter cell gets 1 each of a centromere from chromosome 1 and a centromere from chromosome 2. In the right-hand case here, one of the daughters gets 2 centromeres from chromosome 1, the red one, and 2 centromeres from chromosome 2, the blue one. So these are going to be non-viable gametes for the exact same reason as adjacent 1 is, but this is a rare case because essentially it's 2 non-disjunctions happening at the same time. And so normally what's going to happen is, due to Mendel's second law of independent assortment, about 50% of the time you're going to get this at anaphase 1, and this other segregation possibility is going to happen the other 50% of the time, and very rarely do you get this. So what ends up happening is, even though all these gametes are viable and all of these gametes from adjacent 1 are inviable, you're still going to get out of meiosis in total here 50% viable and 50% inviable gametes over a population, because you're going to get the alternate segregation happening about half the time and adjacent 1 happening about half the time. Yes, adjacent 2 does also result in 100% inviable gametes, but because it is a double non-disjunction event, it is rare enough that it's not going to affect the overall numbers at all. All right? I'm seeing a lot of you not writing down stuff. You should try to do that, like taking notes, even if you don't think it's, you know, I learn a different way or something like that. It's been shown that if you are writing, your brain is engaged. In fact, what studies have shown is that when I'm talking and you're writing, it's impossible for you to write everything that I'm saying. So what your brain is doing is condensing and writing a summary, essentially, of what I'm saying. And by just the act of doing that, that's what helps with you remembering the topics that we're talking about. So also, you know, I intentionally don't write out, you know, I don't have paragraphs on each slide that I just read to you. It's so that you have to take notes. All right, so that was chromosome rearrangement, the whole part of it there. Now we're going to talk about the second part, which is aneuploidy. So remember, the difference between aneuploidy and polyploidy is that aneuploidy is the difference in chromosome number of 1 or more genes, whereas polyploidy is going to be for the entire set of chromosomes. So because we are diploid, we're going to consider diploid correct. So nullosomy is going to be loss of both chromosomes. Monosomy is going to be loss of one chromosome. Disomy is not listed because disomy is just being diploid. That's normal. That would go in here, but it's normal. Trisomy is having 3 and tetrasomy is having 4. Look at the notation. All these have 2N in front of it. So that means you are a 2N or diploid organism, and let's say trisomy. Trisomy is 2N plus 1. That means plus 1 chromosome. Like for Down syndrome that we're going to talk about, it's 2 of everything plus 1 extra of chromosome 21. For tetrasomy, it's 2N plus 2. The number is what you are gaining or missing from one of the chromosomes. Nullisomy means you are completely missing 2 of one of the homologs. If you're nullisomic for chromosome 13, that means you have 2 each of two each chromosomes 1 through 12 and 2 each of 14 through 22 plus 2 X's or an X and a Y, but you're missing chromosome 13 completely. Let's say you were trisomic for 2 chromosomes, like you have 2 chromosomes of 21 and 2 chromosomes of 8, or 3 chromosomes of 21 and 3 of 8. It would not be 2N plus 2. That's tetrasomy of 1. So it would be 2N plus 1 plus 1. That means you have 2N and 1 extra of one chromosome and 1 extra of another chromosome. Make sense? It's just the notation, the way it goes. So how do we go about becoming aneuploid? Well, I mean, you don't want to, but what happens that would go wrong to make you aneuploid? In some way, loss of a centromere. When the chromosomes gets missegregated, it just gets mishandled and then eventually degraded and completely lost. Non-disjunction. We've already talked about that. A non-disjunction event of a diploid organism for 1 chromosome will result in one gamete having 2 copies and the other having 0, so that if they're fertilized, the gamete that had 2 copies will end up trisomic for that chromosome, and the gamete that only had 1 will end up monosomic for that chromosome. We'll talk about Robertsonian translocations coming up. So what I talked about previously and showed in the slides was a reciprocal translocation. Reciprocal meaning that one chromosome gives up a part, a second chromosome gives up a part, and they swap them. You can also get a translocation where one chromosome gives up a part and it just gets tacked onto a second chromosome, but both chromosomes still exist, one's just shorter. Or a Robertsonian translocation, which we'll see is another way you can have trisomy 21 and be Down syndrome, in which 2 chromosomes actually fuse together to make a single chromosome, which is an interesting phenomenon. And that's happened for everybody in this room, in your lineage, leading up to becoming human. We'll see how that works. All right, so non-disjunction. This is a review of what we did in the lab, so get out your stuff for that. You can get your chromosome bags and go through non-disjunction again and see how that works. If you had non-disjunction in meiosis 1, all the gametes are going to have a problem because you start out with one of the daughter cells getting 2 and the other one gets none. And so that's it. One will be trisomic and one will be monosomic when you get fertilization with a normal chromosome, like I said a second ago. So that was a question on the homework as well. In which non-disjunction in which meiosis leads to all aneuploid gametes, and it's meiosis 1. Because you get one with too many and one with not enough, and that's it. No normal gametes are produced. If you have non-disjunction versus in meiosis 2, however, in most cases this will result in 50-50 normal and aneuploid gametes. We had the little little deal we made in the lab, the presupposition that if you get a non-disjunction event in meiosis 1, you're not going to also get a non-disjunction event in meiosis 2 because they're rare enough. And if you get a non-disjunction event in one meiosis 2, for example here, the sister chromatids did not separate. They both go to this cell and nobody goes to that cell. The other daughter cell is going to be normal. So if you have non-disjunction in meiosis 2, you're going to get 50% normal gametes because one of the meiosis 2 divisions is presumed to happen normally. Then you get, in each case, a normal gamete, and there we go. So again, you're going to get one that's trisomic and one that's monosomic, but the other two gametes there are going to be just fine. You can also get non-disjunction during mitosis. It is exactly the same looking as non-disjunction during meiosis 2. During meiosis 2 and mitosis, the sister chromatids separate and are supposed to go to separate cells like this, but if non-disjunction happens, you have that. Here, we have the red, when these two chromosomes, the two homologs line up at the metaphase plate, the red chromosome has normal mitosis anaphase where one chromatid goes to each daughter cell, but here we've had non-disjunction of the second chromosome where both of the blue chromatids go to one daughter cell and then eventually become their own chromosomes. The difference between non-disjunction during mitosis and meiosis is that if you have non-disjunction during mitosis or during meiosis, each of these gametes, once fertilized, is going to give, like this, they're fertilized, is going to give rise to a new organism, every cell of which will have this problem. Every cell here is going to be trisomic, right? This is a germline mutation. It's passed on to the offspring. Here, only the cells that this happened to are going to become aneuploid. So remember the calico cats where X inactivation happens sometime after fertilization where there's just like a ball of cells? That can happen here too, where you can be many, many generations into development and then a non-disjunction event happens. And so your body will have only the cells that this cell was going to lead to having aneuploidy. And so if it happens far enough along in development, like when you're already fully developed and an adult, then it's just a localized thing. So aneuploidies like this lead to a lot of cancers. But that's why you don't, you get like a mole that goes bad or something like that where you have something like this that happens, but it's localized to that area. If it happens early on, we're not going to get a chance to get to Down syndrome today, but if it happens early enough on, we're going to talk about two ways to get Down syndrome, familial and a spontaneous mutation, but there's also a third way, and that is a mosaic like this. So I had a student previously who his brother was a Down syndrome individual, and when they did the test to figure out whether the mutation came from mom or dad, the answer was neither. So what happened is sometime fairly early on in development, one of his cells did this for chromosome 21, and if you are monosomic for 21, you're dead. So the original cell that had this happen, it died out, and that's it. It underwent apoptosis. But now you had a bunch of cells in this kid's body had 3 copies of 21, and the rest had only 2 copies of 21. So he was actually a mosaic, and how early on that happens, so therefore how many of the cells have 3 21s and how many have just 2 of the 21s will have a big difference in how high functioning you are or not. So I learned something. All right, we'll see you guys later. Later today for lab, later otherwise. I have not seen a lot of you guys for, if you did not do study and everything, I think those students who have come in have gotten some good guidance, and maybe you can tell your friends that I didn't treat you poorly or anything. So everybody should come in and talk it over, because I want everybody to do well. If you don't change what you're doing, the outcome is not going to change.